Good morning. My name is Vanessa Ruta, and I'm a faculty member and head of the Laboratory of Neurophysiology and Behavior here at Rockefeller University. It is my great pleasure to welcome you to this morning's Women in Science webinar. I'm especially delighted to host a special program featuring my friend and colleague, Dr. Mandë Holford. Before I introduce Dr. Holford, I would just like to say a few words about Rockefeller for those of you in the audience who are new to the university. Rockefeller was founded in 1901 by John D. Rockefeller, who recognized that the major limitation to preventing or treating diseases was a lack of the understanding of their fundamental causes. The Rockefeller University was created to bridge this gap, embracing the credo, science for the benefit of humanity, which has fueled our efforts for over the last 120 years. Rockefeller laboratories conduct basic biological research to better understand the essential processes of life, health, and disease. The university's discoveries over the past century have enriched basic understanding and knowledge and created many new opportunities to treat, prevent, and eradicate a range of human illnesses. Though many big discoveries have been made here, Rockefeller is actually a relatively small institution. There are only 70 laboratories with no departments. Each laboratory is led by a scientist like myself who reports directly to the president. And by creating this really unique, highly interactive environment with a great clarity of mission and focus, the university allows its scientists, from graduate students to lab heads, to pursue the most audacious research that has the greatest potential and opportunity for impact. Our mission was really put to the test this last year as 25 of Rockefeller's labs quickly pivoted their focus to the basic biology, pathophysiology, prevention, and treatment of the SARS-CoV-2 virus. I think it is indeed a testament to the strength and resourcefulness of our community that we have since advanced innovative ideas and practical measures to influence the course of the pandemic. Recently, Rockefeller entered into a licensing agreement with Bristol Myers Squibb to advance the development of a drug based on 2 monoclonal antibodies discovered at the university in the lab of Dr. Michel Nussenzweig, in collaboration with his colleagues, Dr. Paul Benash and Dr. Charlie Rice. Monoclonal antibody therapies have the potential to prevent people with early stages of COVID-19 from developing severe disease, a critical need as hospitals continue to be inundated by resurgences of the infection. The treatment may also help to mobilize the response to new variants of SARS-CoV-2 recently discovered in the United Kingdom, South Africa, and Brazil, and other variants that are yet to emerge. I'd now like to say a few words about the importance of the Women in Science initiative at Rockefeller. Established in 1997, Women in Science has had a truly instrumental influence and served as a source of support for women researchers at the university. Since that time, contributors to Women in Science have raised more than $32 million to recruit women faculty, fund areas of research that affect women's health, expand the on-site Child and Family Center, a place where scientists' children can be cared for while their parents are in the lab, and importantly, funded more than 240 fellowships for women graduate students and postdocs. So as a faculty member and an alumna of the Rockefeller Graduate Program, I can tell you personally how important the Women in Science initiative has been to me and to my trainees. I consider mentoring our young graduate students and postdocs one of the most important roles I play here at the university, and one that brings me tremendous pleasure. Women in Science fellowships provide essential opportunities for training the next generation of researchers, assuring we capture all the diverse, innovative thinking of young women scientists. And I'm really proud to work alongside Women in Science fellows in my lab. Okay, so now it's my real pleasure to introduce today's featured speaker, Dr. Mandë Holford. Mandë is an associate professor in chemistry at Hunter College and at the City University of New York Graduate Center. She holds scientific appointments also at the American Museum of Natural History and Weill Cornell Medicine. And I think her diverse affiliations really beautifully reflect her approach to science, which spans field expeditions in the oceans of the South Pacific to synthetic chemistry in the lab, or as she says, from mollusks to medicine. After getting her degree in chemistry and mathematics from the City University of New York, Mandë earned her graduate degree here at Rockefeller with Tom Muir, with whom she worked on some very innovative approaches to protein synthesis that combine recombinant biology and synthetic peptide chemistry. For her postdoc at the University of Utah, she chose to apply her biochemistry background to explore the potential of a truly fascinating class of proteins, the small libraries of toxins that make up natural venoms, and went on to study cone snail toxins. So each species of cone snail produces its own unique arsenal of tens to hundreds of related protein toxins that act with exquisite specificity against many ion channels and receptors, serving to swiftly incapacitate the prey of these marine snails. And so while postdoc and now running her own lab, Mandë has been exploring questions related to the evolution, production, and function of these venom peptides in predatory marine snails. Her work has used both taxonomy to identify new classes of protein toxins and biochemistry to map structure-function relationships with the goal of harnessing these toxins for their therapeutic potential and their ability to precisely modulate cellular signaling pathways. So Mandë was a few years ahead of me as a graduate student at Rockefeller, and even in the early days of her career, she was known throughout campus for her energy, for her passion for science, and generosity of spirit. In addition to being focused on research, she's a mentor to young scientists and is devoted to science outreach and education, including developing a course at Rockefeller on science and diplomacy to emphasize the importance of scientific policy. Taking her passion for venomous snails to a new level, Mandë co-founded Killernailscom, an award-winning company that creates games about extreme animal adventures for young science enthusiasts. And so for her many contributions to science education, Mandë has received the prestigious Camille Dreyfus Teacher Scholar Award, a National Science Foundation Career Award. She was also honored as one of the breakthrough women in science by the Howard Hughes Medical Institute and NPR Science Friday. Last year, Mandë gave a TED Talk on the power of venom, and just last month, she was featured in an episode of Nova's three-part series, Beyond the Elements, about the chemistry of life. So Mandë, we are really delighted to welcome you this morning and really look forward to hearing all about your investigations.
Thank you very much, Vanessa, for the very kind introduction. It's been a pleasure to come back, particularly to talk to this audience. Having been a student at Rockefeller, I've attended several of the events and luncheons under the tent, and I look forward to the day when we're able to gather again, once we're all vaccinated, of course, to enjoy those events again. As Vanessa said, I was a student in Tom's lab, and while I was in Tom's lab is where I sort of honed this excitement for doing science. And in Tom's lab, we were looking at investigating, we were using chemical tools to investigate biological questions, which is sort of what I'm still doing in the lab, although we're using different tools. And so this is Tom over here, and this was me way back when I was a grad student. And I just wanted to say that being at Rockefeller as a grad student is a unique experience. It's a culture of excitement about science and a dedication to asking tough questions and then trying to figure out how do we find answers to those questions, as was witnessed by what happened last year with COVID. And so I'm so tickled that my wee ones get to continue in this tradition of discovery and learning as students at the Rockefeller campus, and they're in the Child and Family Center, which thankfully has remained open for everyone since September, with weekly testing being performed by the Darnell lab. So it allows me to drop off my bundles of joy and then go to the lab and engage in doing my science. And so that's what I'm here to talk to you today about. We're going to talk about venom and venomous animals. And so when most people think of venom and venomous animals, they think of them as agents of fear, which makes sense because, of course, some of these animals are lethal and could potentially kill you. But when I think of venom and venomous animals, I think of them as agents of change and innovation. And the reason I think of them that way is because that's how nature has thought of these animals. So this is a family tree of organisms on the planet, and everywhere that you see red indicates a venomous organism. And what we think is that venom is a convergent trait. What that means is that it has evolved independently throughout evolution. And so while we might be familiar with like scorpions and snakes and spiders, you might not know that we also have venomous platypus, we have venomous starfish, the larvae of butterflies and moths are also venomous. And the current thinking in the field is that at least 15% of all animals are venomous. And I think this might be a low estimate, of course, because we haven't surveyed all the animals on the planet. But the take-home message is that venom is everywhere. You can go, they're found in air, in land, and in sea. You can't go anywhere without encountering a venomous animal. And so in the sea is where we find our creatures. And so we work with killer snails. Hidden under the sand is a snail. That orange thing that you see is the proboscis. On the tip of the proboscis, it has a vagular tooth or a harpoon, which is filled with venom that it will then inject into the fish. This is real time. We have not sped it up. And so then the snail rises out of the sand and it will engulf this fish whole. If this is not innovation, nothing is. The fact that a slow moving snail could eat much faster and agile fish is really, really an achievement of nature. And the achievement is in the form of venoms, right? So these snails, they prey on fish, worms, and other snails. And you'll get a chance to see that interaction again here. This is Conus bullatus. We dug it up from the sand. On the tip, you see there the proboscis, and again, it will inject into the fish. Instantly, the fish becomes paralyzed, right? And so what is happening here is that these venoms, they're working very fast, they're very potent, and they tend to be very selective for their target. These are all the ingredients that drug companies are looking for when they're looking for a drug. And so you'll see that although the snail has envenomated the fish, he didn't kill it, and this fish will try to recover, but it's too late. The fish really doesn't have a chance. And so the reason that the fish doesn't have a chance is because venom is not one thing, but several things. I like to describe it sort of as like a cluster bomb. And so in these next few slides, I'm going to try to share with you some features of venom so that we're all on the same page, right? So venom is made up of small molecules, peptides, and proteins. And so small molecules are organic compounds that have low molecular weight. Peptides are made up of a string of amino acids, and peptides tend to be less than 100 amino acids. Proteins are more complex versions of peptides made up of more than 100 amino acids. So each snail, in our lab, we focus mostly on peptides, and each snail can produce anywhere between 50 to 250 unique peptides in its venom arsenal. It's interesting to note that no two snails have the same exact same venom arsenal, and also the venoms are very species specific. So comparing snail venom to snake venom is possible because they have homologous components, but they are not exactly the same things. But they do attack the same physiological targets, right? So all the components in venom, they're mostly working to shut down normal physiological functions related in key areas. So it's like blood, brains, and membranes is the way that I like to describe it. They go for sort of the strongholds that helps an organism maintain its integrity. And they do this primarily by targeting ion channels and receptors that are at the surface of cells, right? So here you're looking at a neuromuscular junction where one neuron is signaling to another neuron. And on the membrane or the surface of these neuron cells, you have different channels, sodium, potassium, calcium channels. And the peptides, they are very selective for these channels. So when they're injected to the prey, it's not one peptide, but several peptides that are acting either to inhibit sodium channels or either to inhibit the channel or to activate the channel. So inhibiting meaning that it's blocking the passage of ions through the channel, and activating meaning it keeps the channel completely open. And so today I'm going to talk to you about the killer snail variety of these peptides, but all of the organisms that we find in nature have this arsenal, and it's what is making pharmaceutical companies very excited, because you have basically this natural pharmacy of compounds that you can apply for looking at manipulating signals. And so that's the third thing, is that venom is very successful. We know that they work. After millions of years of evolution, these venom peptides are become very, very selective, and they can work as triggers for manipulating cellular signals. In particular, they can be used for targeting malfunctioning signals that are often found in human diseases and disorders. And so in my lab, we're studying how venom has evolved over time, primarily in these snails. We look to discover new peptides that are found in the venom arsenals. We characterize the molecular function of these peptides, and we try to identify therapeutic applications in pain and cancer. And so you might be thinking, how do you go from venom to drugs? Is that possible? Aren't venoms like the deadly villain that kills everything? And the answer is yes, it's true. But I think the challenge for us as scientists is figuring out, how do you take something that kills you and use it as a cure, right? And that's sort of the exciting frontier for venom scientists, teasing out the components that are found in the whole arsenal, which if you inject it into a patient would certainly kill them, but looking at those specific components within the venom that are very good at manipulating specific signals and using those to help discover new therapies. And so that's been done, and there are currently several peptides that are, or several drugs on the market that are derived from or are the peptides themselves as they're expressed in venom. And I'll go through some of those with you now. So the first one is from the Gila monster. And the drug that they discovered is Zenatide, which is being used to treat diabetes. And this is an interesting story because Gila monster is a binge eater. And so when they're getting ready to have a big meal, in their saliva, they have compounds that lowers the blood sugar. And the drug that we discovered here is something that lowers blood sugars in patients. Which speaks to learning more about the ecology might help us discover, drugs more efficiently. The second one is from the Brazilian pit viper, and it's called Captopril, and it's used to treat high blood pressure. Captopril was sort of a breakthrough drug, and it not only gave us one molecule, but it gave us a class of drugs called ACE inhibitors, angiotensin converting enzymes, and it was actually the first venom derived drug to be approved. The last one I'm going to share with you right now is from the cone snail, the killer snails. It's the first cone snail drug on the market, and it's used to treat chronic pain in HIV and cancer patients. So what's fascinating about these animals is not only that they're giving us new compounds, but they're also giving us new mechanisms for how we tackle these tough problems related to diseases and disorders. So similar to how Captopril introduced a whole new class of compounds, Preop has demonstrated a whole new way for treating for treating pain. And so, which is very important, as you know, because in the middle of the pandemic, we're also in the midst of this opioid addiction, where currently the number one prescribed treatment for pain is morphine or opioids, and they come with a very real side effect of addiction. And so what Preop has demonstrated is that you can treat pain without having addiction, because the Preop is 1,000 times more potent than morphine, but it doesn't work on the same receptor. It doesn't work on opioid receptors. It works on an N-type calcium channel. So in that way, you can avoid addiction while still treating the pain. But Preop is not a magic bullet. It has to be delivered via a spinal tap because it's not peripherally active. You can't take it as a pill or inject it directly into the bloodstream. You have to deliver it where it's active, and that is in the spinal cord. So as you can imagine, this is a very invasive procedure, and any patient that's in pain doesn't really want to sign up for this. So the use of Preop has been limited to patients for whom morphine is no longer an option. And so in my lab, we wanted to look at, can we find other compounds for, first, I'll tell you the story of treating pain, but can we find other compounds from these snails that can be used to discover new ways of treating human diseases and disorders? And so just to give you a little background on the snails, they come in 3 different flavors. We have cone snails, terebrids, and turids. And cone snails produce conotoxins, which the majority of the work in the field has concentrated in this area. When I started my lab, I wanted to work on terebrids, which produce terotoxins. And this is where sort of my Rockefeller training in grad school kicked in, the idea of taking risks in science. So no one had characterized the venom from these snails before, and we weren't sure what we were going to find, but we wanted to take the risk, and actually we were taking calculated risk. So the first thing that we did a little different from what was done previously is how we collect the snails and how we decided which species we were going to study. So traditionally, if you go to the beach, what was done is you would go and collect as many of the same species as you can find. And so size and opportunity is what drove the discovery of the venom arsenals from these groups. What I wanted to do was look at it from nature's point of view. So really taking what has been done over time to manipulate these arsenals and use that sort of as a roadmap to decide which species and which venom arsenals we would investigate. So for example, and to do this, we start by building a family tree of the snails. So for example, if the Preop drug was found in this snail, it helps us to know what snails it's related to if we want to investigate those arsenals in a more sort of hypothesis-driven, target-driven way. And so this way, evolution and diversity becomes our guide for fishing out the venom arsenals. And so to build that family tree, you have to go and collect the animals, right? So the tree is built from extracting DNA from all of these animals. And so everywhere you see here is an expedition to collect these terebrid snails. There are about 400 of them that are known. And right now we have about 48% coverage. And we've described 69 new species. And while this might sound exciting because they're found in tropical marine environments all over the world, it really is a lot more, it sounds more glamorous than it is. And so I'll just describe, like, for example, to get to Papua New Guinea is a 3-day journey where you go from New York to Hong Kong, Singapore, and then from Singapore to Port Moresby. And every transfer, the size of your plane diminishes, as well as your expectation of ever getting there. And so once you get to Port Moresby, you take another plane off to one of the islands in the archipelago. And then when you go as far as the planes can go, they dump you out into the back of an RV along what some people would call roads. And then you get to the shore and onto a boat, and you're deposited in an island which even Google can't find. But once you're there, you're ready to eat anything and collect anything that's there. And so we did that, and we were able to build the first molecular phylogeny for this group. And as I said, we really are using it as a roadmap to help us identify those lineages that are actively expressing venom in their use for predation or defense. And so we couple that phylogenetic story with transcriptomic and proteomic work to actually identify the arsenal of what is inside of the venom. So what this is basically we're using genetic sequences and combining that with crude extracts, with genetic sequences on the transcriptomic route, combined with crude extracts in the proteomic route to give us this validated database of what we think is actually the compounds that are found in these venoms, so that when we start to screen these compounds, we can have a little bit of faith that we're working with things that are real. And so in applying those peptides to pain, the strategy we came up with is, because Preop is not peripherally active, the first thing we wanted to do was to inject our peptides peripherally or feed them peripherally to in vivo animals. And so we started with a fly assay in which we looked at different sequences of the peptides that were sort of similar to the sequences of the known pain toxins that are out in the market. And then we use flies because you only need small amounts of materials, but you can get lots of information. And so then we would inject or feed the fly to the peptide. So then if we saw a hit in our analysis, already we've gotten past one of the major barriers for the administration of Preop, in that our hit would be something that was peripherally active. And so the assay we developed was a fly noxious heat assay, in which it's basically a hot bath for flies. So the flies are flying away from the hot plate, basically, which is about 46 degrees Celsius. They don't like that. And so if they are not happy there, they'll fly up to the surface. And so we screened several of our peptides, and we found one that was able to allow the flies to tolerate this heat for up to about 2 hours. So this is the time of injection on the x-axis and avoidance on the y-axis. And you see that compared to control, which had no effect, so there was no tolerance for heat. The fly immediately started to fly away once you put them in the bath. We were happy to identify that we do have peptides that seem to have pain activities in the teratoxins, and that the toxins that we have in the terebrids are very different from the toxins that are found in cone snails. They're very unique, as I described in the beginning. And so while doing this assay, but by injecting the synthetic products, will help us to identify phenotypic behavior, we're also interested in looking at identifying the molecular targets. And so in the future, what we're planning right now, or not in the future because we're actually now doing it, is we are synthesizing these peptides with a marker that lights up, so that when you inject or feed the peptide to the fly, we can then trace it and see where the peptide's going to identify the tissues that it's interacting with, and on the surface of those tissues, really identify the molecular targets, the ion channels and receptors. And so when we are thinking about these organisms really as like a natural pharmacy, besides pain, we wanted to look at other sort of intractable diseases that we might be able to apply these peptides to. And the one that we thought of was cancer. And we thought of cancer because there is a history of venom peptides being applied to cancer. And it starts sort of with melatonin, which is a 26 amino acid peptide from the Western honeybee. And it's been indicated in a series of cancer types, from leukemia to melanoma and bladder cancer. And it works by sort of stimulating damage of stimulating DNA damage. This then leads to an activation of apoptosis, inhibition of metastasis, and inhibition of epithelial mesenchymal transfer. The other peptide I want to talk to you about comes from the common yellow scorpion. It's RK1, 14 amino acids long. It's been indicated in glioblastoma and melanoma, and it works by suppressing metastasis and angiogenesis. And the last one that's already known is from the Chinese cobra, CTX3, 66 amino acids long, and it works on breast cancer and primarily works by suppressing EMT. And so in showing you this table, what I wanted to take home is that these venom peptides don't just try to attack cancer from one route. They're using several routes. And these are all the routes in which current companies are looking for potential therapies. So whether it's triggering programmed cell death, so the tumors kill themselves, or preventing the cancers from spreading to different sites through metastasis, or preventing the tumor from drawing blood to itself and development of new blood vessels, or preventing the cancer from becoming migratory and invasive. These animals have peptides that can address all these different stages of cancer development. And that's what's exciting, I think, for us and also for pharmaceutical companies, because you have many chances as which you can attack this disease. And so when we started to look at this problem, I wanted to look at liver cancer because it's a growing threat in the field. The incidence of liver cancer has tripled since 1980. The overall death rate has more than doubled from 1980 to 2016, and it's the fifth most common cause of cancer for men and the seventh most common cause of cancer for women. And the treatments on the market currently are not selective, which is the treatment for most cancer, which is the same for most cancer treatments, right? They do not discriminate between healthy and tumoral cells. And so for the patients, it's really a disaster when you're going through the treatment. So we wanted to see if we could change that. And we wanted to see if we can find a way of using the specificity of these venom peptides to target channels that are specifically in tumor cells and not in healthy cells. So this way we can make the treatments a lot better for patients. So here, this is a liver cell. The green are healthy cells. Purple is the tumor cells, which express a specific channel. And so the idea is if we find peptides that are selective for those channels that are being overexpressed in the tumor cells, we can shrink the tumors and mitigate the cancer without causing a lot of damage to the healthy cells. And so this theory works because ion channels mediate tumor cell survival, death, and mortality, and they're found at all these various checkpoints for how cancer progresses. So whether it's cell migration, cell adhesion, cell cycle control, or invasion and metastasis, you have specific ion channels and receptors that are being expressed at each of these junctions that could potentially be targets for our venom peptides. And so we did a screen of the peptides that we have in our library. And so we screen against various cell types. And here we use cervical, neuroblastoma, prostate, and liver. In blue is the non-treated. In green is the vehicle molecule. And in red is the current cancer treatment, which is doxorubicin. And so what you'll notice is that dox is indiscriminate, right? It is not selective. It works across the board, and it's very good at inhibiting all of these cancers. We were very excited to see that our venom molecule, which is shown in green, while it did nothing or almost nothing for cervical, neuroblastoma, and prostate cancer, it was very effective against liver cancer. And while it's not as good as doxorubicin, it did hit the one check that we were very eager to find, which is something that would be selective, and it was selective for liver cancer. And so when we identified that compound, it was TV1. It was a molecule that we had identified previously. It's made up of about 21 amino acids. It has 3 disulfide bonds, which give it this unique fold for venom peptides. And so we're very excited to know what is the molecular target that this peptide is interacting with, and how is it going to work in vivo. So we injected it into mouse models where they were expressing these tumors under the surface of their skins. and in non-treated animals, which are shown in red. And then we measured the tumor size after an amount of time. So the non-treated animals are shown in red, and the animals who were treated with a high dose of our venom molecule, TV1, had significantly less tumors and tumors of smaller size. And so what is it that TV1 is interacting with on the surface of these tumor cells? Because that's where the peptide's acting, right? So we wanted to identify what the channel might be. So we looked at several channels that might be overexpressed in the cell line that we were using. So in pink are the normal cells, and in blue are the tumor cells. And along the x-axis are different channels that are known to be found in these cells. And you'll see that the TRPC6 channel is one that was highly overexpressed in the tumor cells compared to normal cells. And so that made us think that our TV1 might be interacting, our peptide might be interacting with this channel. So we did a series of confocal imaging in which we synthesized our peptide with a fluorophore that glows green. And we then, this was done in cell lines. And then we injected it into the cells, and we monitored and saw where it was co-localizing. So these are TRP channels, and the blue is the staining of the nucleus of the channel, DAPI. The green is our peptide, which has a fluorescein label on it that makes it glow green. Red is labeling of the TRPC6 channel, which makes it glow red. And then when we combine all of these signals, everywhere that you see red, you're seeing green. So that tells us that our peptide is in the same vicinity and it's co-localizing with these TRP channels. We found a similar result when we looked for another TRP channel, TRPV6. Again, blue is the nucleus of the cells. Green is our peptide labeled with a fluorescein. Red is the labeling of the TRPV6 channel in this case. And everywhere you see red, you're seeing green. And so we think that we now sort of have this working model in which our venom peptide is attacking the TRP channels, preventing calcium from entering. It then leads to inhibition of proliferation, inducing apoptosis, and inhibiting angiogenesis. So again, those routes that cancer use in order to proliferate and thrive. And so we're really excited about these results, but there's still a lot of work to be done because when we tried the peptide in human cells, you needed a lot more of it than you did in the mouse cells to get the same activity. But we do think that TV1 has given us a scaffold for understanding how we might be able to, turn off signaling in liver cancer and how we might be able to turn off those malfunctioning signals that are leading to the proliferation of the disease. And so while we've just started, I wanted to kind of close out by giving you a brief history of peptide drugs, right? And so the first peptide drug was insulin, which is still being used today. It's injected into patients. And for about 40 years, not much happened until Bruce Merrifield, who's a former Rockefeller faculty, discovered solid phase peptide synthesis. And he really just sort of opened the field because now you had a way of making these peptides that was much more effective and efficient. And it was a thrill for me being at Rockefeller because Bruce was still there in Flexner Hall when Tom's lab was on the 3rd floor and Bruce's lab is on the 4th floor. And so what we're trying now with Venomics, I think equally will have a great impact on the field. As we've identified, there's several peptides that are already identified from venomous organisms that are in the field and are effective, and there are even more when we talk about peptides that have been derived from these venomous organisms. But we do have a significant roadblock in that these model systems, there is no model system for characterizing venoms, right? So a lot of what we know about genetics, we've learned through, flies and C. elegans and everything. But for us, we actively have to go in the field and get our animals. We can't breed them in the lab. So traveling is fun unless you're in a pandemic and then you can't travel. So our newest thing is trying to figure out how do we analyze these genes that are found in the venomous animals, and how do we question how they become weaponized for ecological and therapeutic advantage. And the best way for doing that is to develop a model system for manipulating venom peptides, right? And so what we're thinking is we're going to use organoids, which are, organoids are 3D self-organized structures from stem cells that are made to recapitulate the tissue that they came from. And so what we'd like to do is make venom gland organoids. So we were able to sort of stockpile them the way we stockpiled, beans and the way that we stockpile beans and toilet paper during COVID. So while we might not be able to travel to collect new specimens, we can grow them in the lab and essentially have a benchtop version of speeding up evolution so that we can then use these organoids to manipulate the genes that are expressing these peptides to try to make them either more specific for channels that are involved in diseases and disorders, or to make features that allow them to become more peripherally active. Of course, as with all things in science, there is risk, and there are questions about reproducibility and if we can use this venom really and quantity for what we will find in these organoids. But we do think that this strategy might be an effective one where we combine organoids and venomics to help advance, precision medicine. And I'm just going to close out with a small thing of saying that the potential of venom is very bright. The World Economic Forum recognized it as one of the future frontiers of science. And we now have a community. There's enough of us doing it that we're able to gather at Gordon research conferences and talk about it. And I really think it is a transdisciplinary moonshot. This is an area in which chemists, or people trained in chemistry like myself, can meet with biologists, computational scientists, and really try to look for new things that will help us understand how we treat human disease. And not only how we treat diseases, but how genes have evolved over time, which is a basic question in science. And so I hope that I've shown you that these venom and venomous animals are really are not just agents of fear, but they're also agents of change and innovation. And with that, I thank you, and of course, all the members in my lab and all of our collaborators who help us do this work. And a specific thank you to the Women in Science Initiative at the Rockefeller University for the invitation to come and share our work with you today.
Thank you so much, Mandë, for your fascinating presentation and how you've been able to leverage biodiversity to identify new therapeutics. So we'll now turn to some questions from the audience. Just as a reminder, you can submit a question through the question and answer function at the bottom of your Zoom screen. We already have a couple questions, so maybe we'll just begin. Muffy Potter Aston asks, are there any potential anti-aging benefits from use of venoms in stopping the cellular destruction of skin and other bodily functions?
Oh, yes. Yes. This is also a growing area. In addition to the therapeutic appeal, there are lots of companies that are using snail venom in particular for anti-aging and anti-wrinkling remedies. And it works very similar to Botox, right? In that it's sort of it helps to paralyze the cells for a little bit so that you get your nice glow. And this is definitely a growing area of interest. It's not something we're pursuing in the lab, but more and more, lots of labs are looking at that question.
Okay, great. There's a technical question from Charles Ju, who asks, since the venom snails mainly prey on fish, the question is, he wondered if zebrafish would be a better assay than flies for the venom peptides. So what?
Sure. So they prey on fish, worms, and other snails. And in particular, our terebrids, they are worm hunters. And so we can use a worm assay, we can use a fish assay, and a zebrafish might also be a good option. I bring out that the drug that's on the market comes from Conus magus, which is a worm hunting cone. So it really doesn't seem to matter what assay you're using in. In the end, we want to find things who are effective for humans.
Great. Mary Lou Silo asks, is there enough venom in the world for now and the future?
Oh, there's more than enough venom. If you think about it so if we have about 400 species of terebrids, and each of those expressing 250 different venom, just peptides, not talking about small molecules and proteins, then we have over hundreds of thousands of venom molecules that we can potentially explore. And this is what is exciting to pharmaceutical companies, the idea that you have all of these natural products that they know work, right? Because there's no way a snail should be eating a fish, but it absolutely is, and it's eating it because of these molecules. So when you think of it then on the scale of all of the venomous animals out there, we're talking about millions and millions and millions of potential bioactive compounds.
Great. Carolyn Patterson asks, fascinating. She says, thanks. Is venom currently being used by clinicians for all types of pain or just for cancer?
It's been used for all kinds of pain. So the one that I showed you, Preop, is for chronic pain in HIV and cancer patients, but there are other like different inflammatory pain that it's also being used for, not from the snails and not Preop, but other peptides that have been identified for pain treatment. But they're not quite at the drug level. They were still in clinical trials.
So I had a question that was related to the kind of abundance of venoms on the planet, which is, it seems like you can tap into the importance of biodiversity, and there's so many interesting, sources of venoms in different organisms that you focus on snails. Are other people working on parallel efforts to look at venoms of other species like snakes or spiders in a similar way as you are doing? Are there parallel efforts in other species?
Yes, absolutely. The field is sort of dominated by snakes. This is the gold standard, which kind of makes sense because snake bite is still a real disease, and it's a neglected tropical disease, in fact. And I think thousands of people are dying from snake bites a day. People die from snails, but not so much. And so most of the field has been dominated with snakes doing parallel work in terms of discovering what these peptides in snake venoms can do. And so we do have a rich history that we can cull. So when I talk about doing calculated risk, we are not jumping completely into the unknown. There is a history of what we can cull from when we want to apply it to our Herbert snails.
Right. There's an anonymous question. Can you explain the difference between a poison and a venom?
Oh, yeah. Oh, very good question. This is a great question because a lot of people get confused by this. So the way they differ, there are three ways in which they differ. One is in delivery. So something is venomous, it has to pierce your skin, right? It has to actively be delivered into your skin. A poisonous thing does not have to do that. If you think of poison ivy, you rub it on your skin and you can get affected. The other is the venom has to be expressed by the organism. So it has to be an endogenous expression of the molecules. For poisonous things, they can acquire it from their environment. So if you think of poison dart frogs, they eat certain things in their diet that enables them to, secrete an alkaloid on the surface of their skin that is very poisonous. But if you don't feed them, the thing, then they won't make the poison. So that's the big difference. And then the third is the components within the venom arsenals. So mostly venom is made up of, as I said, small molecules, peptides and proteins, whereas most poisons are made up predominantly of small molecules and specific small molecules called alkaloids. So there isn't a scientific definition that separates venomous and poisonous animals, but most people just call them all poisonous.
Okay, thank you. So there's another anonymous question, which is asking about this very cool kind of system you spoke about at the end. Is what are the advantages of using organoids in scientific study? Maybe you could go a little bit into more detail about that.
Sure. So the advantage there is because you really have on the benchtop a way of manipulating genes. And so if we want to, peptides are expressed from genes. And so if we want to manipulate how the peptides are really working, it helps us to go back all the way to the fundamental expression level of how they're being expressed. And so what it enables you to do is basically to tinker. So you can tinker and ask questions and get a response. We can't tinker and ask questions with the snails themselves because we have to kill them, sacrifice them, basically. And also we can't breed them in the lab. So every time we need to get a new supply of snails, we'd have to go on an expedition, which is not a bad thing, but it does slow down the work that you can do. Having organoids is a way of replicating that venom gland, and then we can then tease out and ask fundamental questions about, how these genes have evolved over time. We're thinking that these genes were co-opted from housekeeping genes. So like things like making cell walls and things that make ribosomes, and then they came into the venom gland and got weaponized. So it was like, how did we weaponize these genes, and what is the process that led to it? And can we maximize that for identifying things that might have therapeutic appeal more effectively and more efficiently, and also for answering important questions in science.
Okay, so Lulu Wong asks, with so many different venoms in nature, are we early in the discovery of other applications of venom in medicine? And what do you think are the greatest the areas of greatest potential so far?
Oh, absolutely. I'm with you. I'm with you. I'm excited about what we can do. And I think we really have just touched the surface of this field, because predominantly the way that the field has been studied is from a natural products drug discovery point of view, looking at the ecology of the animals and using that to help give us sort of, hypothesized directions, or more sort of sort of focused directions of copying what nature has done is really the new twist. And that's sort of what my group is doing. We're letting nature lead the way. So combining the ecology with the chemistry to study really important biological questions is where I think the field is heading. And new tools like organoids would enable us to really sort of get there faster, because again, as I said, we'd be able to manipulate those questions within the lab.
Catherine Overlock asks, given that venom causes paralysis and cell death, what are the chances that there is an unintended damage to other parts of the body? Do you really understand how isolated the impact will be?
Oh, that's a very good question too. And so the reason that people are also excited about these venoms is that they tend not to be leaky. So in drug development, that's the big question, right? How you can have a drug and limit the side effects so that you don't do damage to other areas. But these peptides are so specific for their target. What we find is that they have minimal amounts of side effects. And so we're hoping that that continues as we discover new drugs for this area, that we can really minimize the side effects. But you can never really get rid of all of them, but you can certainly minimize them.
There's an anonymous question, which is, how is climate change affecting the biodiversity of venom-producing organisms?
Climate change is having a big impact, and we have to, as you, this is a smart audience, so you're all aware that climate is changing the way that we live our lives, and it's also changing the lives of these venomous animals, because they're found in the air, in the sea, and on land. All of their habitats are being threatened by what's happening right now. And so for us, when we are talking about our marine organisms, it's really a question of trying to save the oceans, right? And so figuring out how we can mitigate what's happening to save the oceans. And one of the ways that we're doing it, again, is through conserving how we, trying to conserve the organisms when we go to collect. So remember that tree where the black line indicated the species with a venom apparatus? When we go out to collect, we really are mindful on how we're collecting these organisms, although our small collection trip really pales in comparison to what's being done on a large scale to affect the planet. And so it's a real question, and I don't have answers for that, but it is a real consideration, because we're destroying things before we find them, basically, right? And if we have this natural pharmacy that we know of compounds that work, why would we try to destroy it?
Yeah. So there's another anonymous question. Thank you. Your talk was very enlightening. Do you have an idea or hypothesis about how these species improve on their venom peptides' affinity and selectivity without harming their own cells?
Oh, yes. This is a very good question. What we think is that inside of the animals, the venom is packaged into little containers, vesicles, that prevents them from harming the animal itself. But we do know that they can harm other species. So snails can feed on snails, so that the venom can work on other killer snails. But no snail has been that we've aware of can envenomate itself. And we think it's because the venom is packaged in these vesicles. And then once they are ejected into the prey, the vesicles burst and the arsenal is released.
So maybe I can actually, you've told us so much about your interesting and incredibly cool and fascinating science, but maybe I can ask you a little bit more about your outreach efforts. Can you tell us a little bit more about the company Killernailscom and how it's encouraging kids to develop an interest in STEM? I know how important education is to you.
Oh, yes, absolutely. So this is a fun project. We started a company a few years ago called Killer Snail. It's an ed tech company where we use the extreme biodiversity of nature, right? Because these are amazing stories. You see a fish eat a snail, you're immediately, enthralled. to address how we teach STEM to kids. It's not a question of what we teach, it's a question of how we teach. And in the company, we're using games. So we sort of want to meet the kids where they are. And every child loves to play a game for the most part. They come open, they come ready, and they come, sort of either a little bit competitive and wanting to win. And so we thought that would be a good medium for how we change how science is being taught, because the problems coming at us, like the previous question about climate change and the pandemic that we're in has witnessed, that the problems that are coming at us have science and technology at their core. And so it's essential that we get our kids up to speed on the critical thinking and analytical abilities that a STEM education can provide. And while we don't want everyone to become a scientist, well, they can if they want to, but we do want to sort of permeate that sort of critical thinking throughout different career STEMs. So our company makes games for K through 12, and our first game, called Assassins of the Sea, was made in collaboration with the American Museum of Natural History. And we really are just trying to change how STEM is taught, because when you think about games, there's no anxiety there, as opposed to when you gave a kid a textbook and you say, well, here's the periodic table, go figure it out. In our thing, we're using AR and VR and tabletop games to bring the periodic table to life so that the kid is excited about what they're learning. And in the middle, they're getting some science.
It's great. There's actually a related question about your perspective of being a woman and a person of color. How actually how you capture diversity and bring it into your career, and how it actually has affected your career personally, your trajectory.
Well, I think diversity is very important in science, because for sure, science is about the questions you're asking. And so if the same people are asking the questions, then you're going to get similar questions. And so the more voices that we bring into science, I think the better it is. And for me personally, being able to demonstrate that, I'm a big believer of the see it, be it philosophy. And for some people, you need to have a goal. And if that goal doesn't look like you or sound like you or haven't had your lived experience, it's hard to imagine being that goal. And so while I don't thrive in the idea of being a role model, I don't think anybody stands up for that really, I do realize the importance of the position I'm in and the importance that it has for encouraging other girls of color who are interested in science to let them know that it is possible and it is something that they can achieve. And we need those voices. And it's not because of a feel-good moment, but it's more about how science advances. Science advances by asking interesting questions. And we can only ask interesting questions if we broaden. The more interesting questions we can ask, the better. So if we broaden the scope of who gets to do science, we really will advance the science enterprise as a whole.
Yeah, well, I think your seminar, made it very clear that you really are a wonderful role model for, scientists and non-scientists alike. There was a question I just wanted to repeat, that the name of your company is Killernailscom. So I'm sure that hopefully people can find it if they're looking for it on your website. Is that true? Is there a way?
I don't know if it's on my website, but it has its own website.
Okay,
it's a legit company outside of the lab.
Okay, so maybe we'll turn to one last question as we draw close to the end of the hour. Why does the venom not harm the snail in the process of digesting the envenomated fish? So
why does it not
harm the snail once the fish has been envenomated? How come it does not harm the snail as they start to digest it?
Oh, this is an interesting question. And in large part, I think it's also why venom drugs are so good, is because they are able to be chopped up and they clear the organism. They don't stick around for very long, right? So they get to their point of action, do what they have to do. And because they're made up of amino acids, there are things in the body that break up those amino acids and shuttle them away for waste. So I think it's in part because the venom are able to be chopped up, that the fish is not a lethal component for the snail to eat.
Okay, so I think since it's drawing close to the end, maybe it's time to wrap up our program. So I'd just like to thank you again, Mandë, for such a wonderful and illuminating presentation and for being with us this morning. And I'd like to thank you all in the audience for tuning in and asking really such great questions. Before you log off, please make a note of our next Women in Science program on Wednesday, March 24th at 4 p.m. This webinar will be the second in the new Women in Science Entrepreneurship Fund Speaker Series. This series features talks with women leaders in biotech and pharma with the goal of encouraging more women scientists at the university to consider the multitude of opportunities they have in moving from bench science to the development of therapeutics. The upcoming talk will feature Dr. Brandy Hauser on the topic of demystifying the therapeutic development ecosystem, a roadmap of what you will need to know. We hope that you will attend what promises to be really an insightful talk, and to register for the program or to make a gift to women in science, please visit the website, which will appear on the closing slide. So thank you again for joining us. Please be safe and have a wonderful day.